Arthritis Ireland has been working with patients for about 32 years. We celebrated our 30th anniversary 2 years ago. And I think we have a reasonably good idea at this stage what it's like to live with arthritis. The pain, the fatigue, and the social isolation that results from that, the loss of work, and the emotional impact. Now, many services we've developed over the last 10 years have been aimed to try and ease that. In fact, we've spent over about €8 million in the last 8 years delivering services to people in the community. Our self-management program has been delivered to nearly 4,000 people who enjoy a better quality of life, fewer GP visits, hospital visits, less dependency on NSAIDs and painkillers. They exercise more, and in fact they volunteer more. Most, if not all, of our branch network, all 22 branches set up in the last number of years, are born out of our self-management program. So they're of huge value from a volunteering perspective. We've nearly 25 walking groups, support groups all around the community, and a significantly wide information suite of booklets. And I think we've raised the awareness significantly over the number of years. We've still a lot of work to do on that. However, that's not where we end. If we ask every single one of you in the room who has arthritis, or all our members, is there one thing we can do for you? Every single one of you will say, can you find me a cure, please? And that's blatantly obvious. 4 years ago, Arthritis Ireland embarked on realising a dream that was set out by our former chairman, the late, great Professor Barry Bresnahan. Many of you would know Barry. He was a personal friend of mine and a fabulous rugby coach where I first met him. And then I ended up 20 years later working with him. So the moral of the story is never burn your bridges. You never know who you're going to meet. That dream really looked at placing Ireland on a competitive circle within rheumatology around the world. It also wanted to address gaps in education in our graduate and undergraduate levels. I'm really delighted to report that Arthritis Ireland is realising that dream. And with the first appointment of an Arthritis Ireland Chair of Rheumatology in UCD, we are now starting to see that dream come true. We would hope very soon that we will have an announcement in TCD as well, on a complementary chair. And when I first talked to the guest speaker tonight, Professor Gerry Wilson, I told him about our dreams and our aspirations, and that was to create an Institute of Rheumatology in Ireland, funded by Arthritis Ireland, that would hopefully one day find a cure, and certainly in the meantime allow us and help us to understand arthritis a lot more. But of course, typically Gerry turns around and says, well, I've got a better dream and a goal, and that is to put Dublin on the map as a centre of excellence in rheumatology in Europe. So I kind of like his thinking already. I'd like to thank everybody who was involved in getting this far. The Atlantic Philanthropies, headed up by Chuck Feeney, was a significant and is a significant funder behind the chairs of rheumatology. Our industry partners, AbbVie, Bristol Myers Squibb, Merck Sharp Dome, Roche. One of our ex-board directors, Michael Roden, who was a significant contributor to the chair. He's not here tonight, unfortunately. Michael's father was a founding member of the Irish Society for Rheumatology. And of course, many of our volunteers and branch network really got behind this, from climbing mountains, cycling bikes around Ireland, to holding fashion shows everywhere they could. So there's been a diverse source of funding for this, and I'm delighted to be able to introduce to you tonight the first Arthritis Ireland Professor of Rheumatology, Professor Gerry Wilson.
Good evening. So let me just, it's working great. Do you want me to hold something? No? Okay. Right. Well, that's one of the big worries of tonight over the projector has worked as my computer. So it's great to finally be here in Dublin and back in Ireland. And John asked me to talk tonight about sort of my plans. And so over the next 20 minutes, I'm going to do that. And let's keep it informal. If I start to ramble or you want to ask any questions at any stage, just ask away, okay? Just let's keep it informal, which is best. So I'll just give you the outline of what I'm going to do over the next 20 or so minutes. I'm going to just tell you a bit about me, just in one slide, and what I've done up to now, and my research interests over the last 20 odd years. What I've done mainly when I've been a bit in the States, but mostly in England. And then I'm going to just touch about what John was talking about there, actually the current standing of research in rheumatology in Ireland, and actually show you how actually good it is. Actually, I knew that anyway before I came back, that Ireland is one of the great places for research, particularly in Dublin, two big universities, big teaching hospitals. And I'll just show you the metrics, as they say, as to how we measure excellence and how good Dublin and Ireland is at actually doing important research in rheumatology. And we hope to develop in that. And I'll just go through some sort of future plans that would try to develop that further. So this is me. So I qualified in Queen's in Belfast some years ago. And then I did a arthritis, it was Arthritis and Rheumatism Council in those days. And it's now its third change of name recently to Arthritis Research UK. So I got a 3-year fellowship to do a genetic study on the gene that makes TNF. And I spent 3 years doing that. And I was in the lab of Gordon Duff, who was the guy actually who discovered TNF in joints in 1987. He first reported a funny molecule, a funny protein in joints. And when he originally sent the papers in for publication, they were rejected. And some of the reviewers asked why he was looking at a protein that was called tumour necrosis factor, and should it not be more to do with cancer. It's actually interesting how that got its name, TNF, this great, this molecule that's so important in rheumatoid arthritis. And it's got that name that is in a different field. And it's really not so important in cancer, but it's really important in controlling inflammation. So I did my PhD, got it through in 95, and then I got a travelling fellowship to go to Stanford University, again funded by Arthritis Research UK. And what I did there was to look at how the genetics of TNF gene affects how it works. And lots of people, when I sort of published my PhD, looked at what I'd found, and there were a variant that 2% of people in here will have in the TNF gene. And if you are unfortunate enough to get malaria, you're much more likely to die from malaria, for example. And so when I went to Stanford, I continued to looked at really how the polymorphism, how changes in the gene that makes TNF actually makes the gene much more active, and how that can make a lot of diseases much worse. Mainly, actually, it turned out to be infectious diseases rather than rheumatoid. And then when I was in Stanford, I had about 6 months to complete my clinical training, and I was approached by Oxford, and I completed my clinical training in Oxford. And then we moved around the country, so we moved from Belfast, and we'd moved house about 9 times in 6 years, and I had 3 kids less than 6 years of age. And so my wife, who was born in Oxford, didn't want to move there, so we moved back up to Sheffield. And so I was a consultant and really an academic in the Sheffield Medical School from September 97 until the 30th of June 2013. And then I moved to Dublin. And so what have I been doing research-wise over those times? So this is Voltaire, who was a French philosopher, wrote this over 200 years ago. And it's really quite relevant to a lot of what we do. And the quote is said, this is in 3 parts, as you can see. The first part is actually used to be right, but now it's wrong because most doctors are not men. It's 60 to 65% females in most medical schools, actually, so that's wrong. But most of my research is to do, and I'm going to show you, is to do with the second and third bits of this quote from Voltaire. And that's, so, to cure diseases of which they know less, in humans of whom they know nothing. So we've really in rheumatology moved, as I'm sure a lot of people here know, and Doug Veal up there knows, 20 years ago when I started doing, 25 years ago doing rheumatology, we had lots of drugs, we didn't know how they worked, we still didn't know how they worked, gold and histamine. Gold's a good drug, but we really don't have any idea how it works. And we've, because of really good science, very top, cutting edge science, we've been able to work out the actual key molecular drivers that cause inflammation in joints. And that has led to all of these great drugs that we now use, inhibiting TNF or IL-6, or knocking down the cells that we've worked out are really important in driving inflammation, such as rituximab that knocks out the B cells, or abatacept, which inhibits communication between cells, the inflammatory cells in the joints. So really basic science in rheumatology over the last 25 years has really had profound effects on how we treat inflammatory joint diseases. And I'll show you some of the a population of patients I had that I've done a lot of research on in Sheffield, and we recruited them prior to anti-TNF coming on the market. And I'll show you what rheumatoid was like and what it's like today. So the other bit of this quotation is about in human beings of whom they know nothing. And what's that to do with is the fact that everyone is different. And that's to do with a whole lot of things. It's to do with your genetics, and it's to do with where you live, your environment, what you do. If you smoke, if you drink, you get exercise, but your genetics. Okay, so those, we're really working out now how the environment and genes make people respond differently. So rheumatoid in one person is very different from rheumatoid in another person. Heart disease is the same. Heart disease can be a mild form of angina, gives you a bit of chest pain when you walk up a hill against a cold wind, or it can present in 20s or 30s with a fatal heart attack. Any disease, there's a huge variability in severity. And so I'm going to talk to you about that, trying to work out, which is really key in many conditions. At the time when someone presents with a condition, trying to work out who's going to get a severe form of the condition. Because what we can do nowadays is to treat with very effective drugs and switch things off and prevent damage. And that's really what we're trying to do. So the next slide is the first experiment I ever did in my PhD. And it's really quite simple. I took blood from 20 individuals and stimulated the blood cells, and what you see in this graph is the amount of TNF that's produced. So some people produce very little in the way of TNF. Let me see if I can, down here. So just on the left, let me move away. So just here, these individuals, healthy individuals, produce very little in the way of TNF. And you go out here, do exactly the same, take the same cells from these healthy individuals and measure the amount of TNF. There's a 40-fold difference, and we did this on a monthly basis in the same individuals, the same volunteers. And so it's just a bit of human variability, but you can just imagine how this may impact on if you get any form of illness, how it may impact on how severe the illness is, whether it's an infectious illness or it's an inflammatory illness. You may also, it may also be important in if you determining if you're going to respond to anti-TNF agents, for example. If you produce very little in the way of TNF, well, perhaps you're not going to respond to giving anti-TNF, and you should perhaps go on to another drug like rituximab. So my PhD actually looked at genetic markers in the gene that cause the variability in TNF production. And that really is very important for lots of conditions, as I said earlier, particularly if many of the major infectious diseases. But also we have evidence on this variability affects how you respond to anti-TNF agents if you've got rheumatoid arthritis. So the two bits of the research I'll just briefly mention. Patient stratification. Not all patients are the same. No two patients are the same. And rheumatoid in one patient is not the same as rheumatoid in another person, generally. And the other thing is, big area is therapeutic targeting, getting the right drug to the right person, okay? Making a personal decision on what is the best drug for you, rather than just, as we sort of still do, it's really an art form. It's a decision, and it's a decision really that's left up to the patient and the nurse generally. But to get a bit more in the way of science and be able to predict the right drug for the individual patient. So I'm going to talk on both of those. So let's just talk about working out who gets severe disease. So I'll just show you one of the standard ways, and I'm going to talk about rheumatoid. That's where I've done most of this research. So one of the ways you can assess rheumatoid, as you know, is to take x-rays of the hands and feet. And we can score the x-rays. There are lots of different ways of doing that. And one of the standard ways we do it is to take x-rays of the hands and feet and measure what's called the modified Larson score. So there are 32 joints, and each joint is scored between 0 and 5. And you can see 0 is a normal joint. And if you just move across, increasing damage to grade 5, where there's no joint space left, so the cartilage has disappeared. And you can see the nibbles out of the bones, the erosions, what we call erosions on both sides. So this is a measure, a standardized, validated measure of X-ray, of severity of rheumatoid arthritis. And so if you just work it out, it can go from 0 to 160. So if all the joints are damaged, that's 5 times 32, which is 160. So when I worked in Sheffield, I was able to recruit about 20% of our patients. So Sheffield's about 650,000 patients. So we had about 6,000 to 7,000 patients with rheumatoid attending clinics. So I was able to recruit just over 1,000 patients into a study. And what we did was to do x-rays, and I got one of the radiologists to score the x-rays. And this was patients were recruited between 2002 and 2007. So they're not early rheumatoid. Most of these patients had rheumatoid long before anti-TNF came on the market. So here you have disease duration and X-ray damage on the Larson score, and you can just see the variability in the X-ray damage in these group of patients. So these are 20%, about 20% of the patients attending clinic in Sheffield. So if you just focus in, there's year 12. So these patients had rheumatoid arthritis for 12 years, and look at the variability. That unfortunate person at the top, who's a Larson score of about 130, and down to the bottom, a lot of individuals with no joint damage at all. And actually, what's interesting, as you can see, there's a patient with what we say is rheumatoid arthritis for 57 years, and there's no joint damage. So clearly you wonder, is that rheumatoid arthritis? But you can see the variability in how bad it is. So just take two of these. This person after 4 years has lots of damage, a Larson score of around 80. And this person, a Larson score of around 10 after 17 years. And these are the x-rays of those two individuals, those two patients. So this is the first patient who has rheumatoid arthritis. The x-ray was taken in 2005, so had rheumatoid arthritis for 17 years. And those are the x-rays of the hands. And you can see the x-rays of the hands are normal, no damage. This patient has rheumatoid arthritis. Patient lowered in the x-rays from this patient, you can see the severe damage to the wrists and the MCP joints, the knuckles. Unfortunately, that patient had only rheumatoid for 6 years. So you can see how it's different in its severity, and this is really key. And what we really would like to be able to do would be to predict when patients present and work out who is going to go along the very aggressive route. Because clearly if we knew that, we might then intervene with anti-TNF agents. And perhaps in the patient in the lower, we may not actually need to use any aggressive treatment at all if there's not going to be any damage, joint damage. So these are really important things that we need to be able to predict. We need to stratify patients. We need to know potentially who's going to develop severe disease. And so lots of us are doing, Doug Veal's doing a lot of this work as well. What we do is measure things. Look at biomarkers, which are just things that we can measure biologically, whether it's genetics, whether it's things in the blood, proteins in the bloods. And of course, some of these biomarkers, I'm sure you're all aware of some of these biomarkers, the measures of inflammation, CRP and ESR, and the immune system, rheumatoid factor and CCP. So you're aware of some of these, and we've been looking, so part of my research over the past 5 years has been looking at the genetics of severe X-ray damage in rheumatoid arthritis. And I'll just show you one. So we just published this this month in Arthritis and Rheumatism, and it's a paper where we looked at a genetic variant on chromosome 5, which had been shown about 3 years ago to be important to increase the risk of developing rheumatoid arthritis. And so what we did, we looked in 3 populations. The Sheffield population I described, Leiden and Leeds University. So I looked in my lab, we got DNA from about 1,800 patients, and we essentially looked to see if there was this genetic variant correlated with the amount of X-ray damage. And the graph here is from the Leiden cohort of patients with early arthritis who presented and were followed up with X-rays over 7 years. So they had X-rays every year. And we looked at the genetic variants. So there are 3 types, as you can see. And what we see is this genetic variant is associated with X-ray damage. So if you have the variant that makes you more risk of developing rheumatoid, it actually is making you at risk of developing a severe type of rheumatoid with more X-ray damage. Now the thing about genetics is it's easy, they're easy to measure. The genetic things don't change. You're born with your genetics essentially. And we now know that about 60 to 70% of the variability in all those dots that I showed you a while ago is genetically encoded. So the major thing that drives the differences in the X-ray score, the total amount of X-ray damage, are actually genes. And so we're now trying, a group of us, to comprehensively map the genes that are causing more severe X-ray damage, because we could use that information at time 0 when patients present with rheumatoid arthritis to predict who will get severe disease. And if we can predict who will get severe disease, then we can try to switch it off with anti-TNF agents at time 0. And hopefully funding agencies and governments will see that that's cost effective. And that takes you into a whole different thing of pharmacoeconomics, and they'll want to do the costings and work those things, which I suppose are reasonable things to do. But I think it will be relatively easy to get that done. So genetics is one way that gives important insights into who will develop severe types of rheumatoid. So the other thing that I've been looking at is the variability in drug response. Between patients, as you know, about 30-35% of patients who are started on anti-TNF don't get a good response to that, and we have to switch to another anti-TNF agent or to another drug. I mean, TNFs, these new treatments have revolutionized the way, as I said earlier, they're completely, lots of you know, they've revolutionized how we treat and just outcomes for patients. And I hope no one's, you're a bit after your tea or dinner now, so you may get a shock as to what I'm just going to put up for you here. And This is the sun. So the sun, actually, Melanie was one of my patients, and they did a feature a couple of years ago, well, it's more than a couple of years ago actually, where they were looking at chronic diseases in young people, and they did this feature on Melanie. And as you can see, so Melanie presented, Stephanie presented with severe rheumatoid with X-ray damage, and it was switched off with anti-TNF agents. And she didn't become a rheumatologist because she ended up becoming a dentist, actually, which is interesting. She was able to become a dentist, and I have 3 or 4 patients who are dentists, and you can imagine how important it is to be dexterous. And immediately on an anti-TNF. In England, in Britain, you've got the NICE criteria, and you have to treat with 2 DMARDs for 6 months, one of which must be methotrexate. And so really you shouldn't be able to put people on anti-TNFs for at least a year. But with X-ray damage at time 0, I sort of knew this was going to be a bad variant, and so we started Stephanie on two DMARDs, and then got her on anti-TNF after 6 months. And so now she's able to be a dentist. And you can just imagine the outcome 15 years ago, prior to biological agents. So these are great molecules. Surprisingly well tolerated. The anti-TNF agents, all the biological agents. The downside clearly is the cost, and the fact that about a third of patients started on an anti-TNF or any really of the biologicals don't have a satisfactory response after 6 months and need to change. And of course the other thing is that we've got so many different biologicals these days. So we've got the 5 anti-TNFs, IL-1 receptor antagonist, which isn't so good for rheumatoid. It's good for some other conditions. And then the IL-6 inhibitor, which is tocilizumab. We've got rituximab. See, I can't even remember some of the names. And then abatacept. And there are other ones that will be coming out. They're in the process of being approved. So there will be lots of new ones coming out. The other thing is that the orally based new molecules for inflammatory joint diseases, which there are some of them available at the moment. They haven't probably progressed as rapidly as we thought 2 or 3 years ago. And so the biologicals that are given by injection will be around, but they're great drugs. But there is this problem that they're very costly. About a third of patients don't respond to them and need to move on subsequently. And some patients have to, as you know, go on to 3 or 4 or more of these biological agents. And if they're not responding to a biological agent, clearly the risk is the rheumatoid is active, and there may be ongoing damage to the joint. So we'd really like, for those various reasons, those reasons, to get the right drug to you using science and using a rational approach to get the right one for you. And so this is another thing that we've been doing. We in Britain, clearly, one of the big things is that there's a register. So all patients started on a biological for rheumatoid, for example, where you have to be approved by NICE. And part of the agreement was that if the NHS was funding these agents, patients became part of a register. Okay, so all patients who started a biological in England, they're registered with the BSR Biological Register in Manchester. And so I get lots of letters about patients every 6 months where I'd have to fill in the DAS score. And so it's a great resource for doing research into ways of predicting who will respond. And so we set up a study, the 4 of us set up a study, and we've been collecting samples from patients who have been starting anti-TNF, rituximab, and all the other biologics. And we're up to we're actually over 4,000 patients that we've got DNA and lots of samples, and we've got great clinical scores in them. And we started to collect lots more from patients starting. And we're collecting samples every 6 months in some patients. And we've been doing lots of studies, which I won't go into, but we are identifying ways of predicting who will respond best to individual anti-TNF agents in the genetics, in the antibodies that we measure, CCP antibody, for example. So CCP antibody, actually, patients who are CCP antibody positive actually tend to respond better to rituximab. So we do use ways, that's a test that we use at present to predict which drug we will use. It's not particularly effective, but it does give some idea. And so we tend to use Ritux in CCP positive patients as a second line. This was what we did in Sheffield. And if a patient was CCP negative, we would tend to use tocilizumab, which is anti-IL-6, as a second line agent rather than Ritux. So hopefully in the next 5 to 10 years, we will be applying science to make the decision as to what's the best drug for each individual patient. So, rheumatology research in Ireland. I'm just going to tell you, give you some what we call the metrics of how good it actually is. And so, what this shows you is I looked at the number of publications in the two big journals in rheumatology, which are Arthritis and Rheumatism and Annals of Rheumatic Diseases. So those are the big ones that all the academics want to publish in. I know Doug Veal's very keen to publish there, and so am I. And it's, you know, these are ones that as academics you have to be publishing in. And so I just did a search based on the city of origin of the papers in these two journals. And the big cities, and you can see the numbers for yourself. How well Dublin does. It's way ahead of most of the big cities in England. I mean, way ahead, despite not really having a focused approach with academic positions over the years. Dublin has been really good and is well known and well respected for its research. And actually, I'll just show you, I don't know if hopefully this will project. This was just published last month in one of the journals. And so this is how good, how productive the countries are. 35 of the richest countries in the world. And you'll see that Ireland, in these two metrics, so on the right, that's the number of times the papers are cited per billions of GDP. It's one of these fancy measures. But actually Ireland is 8th in one and 10th in another. And if you looked on, we're way ahead of Germany, France, the US in our research productivity. This is rheumatology, actually. So, and that's a testament to many people here, Barry and Oliver, Fitzgerald and Doug Veal, Ursula, Fearon, lots of people. It's a great place. And actually the other great strength is we talk about immunology. A lot of rheumatology is immunology. And if you do the same for immunology, actually Ireland is number one in the world. And probably Dublin is the city of the world for immunology, which is phenomenal. So that's another big attraction to me about Dublin, is there is a great research base already. Lots of people interested in doing really good research. And it's what we call translational research. It's research that involves patients. And that's been ongoing for years. Very clinically relevant studies. So research plans, what I hope to do, and John mentioned this, we hope that over in the next 12 months we'll be able to appoint to the chair in Trinity College, and that will probably be, I would imagine, focused in immunology. And I would expect, given the reputation that I just talked to you about of Dublin and immunology, that we should hopefully be able to attract a very good, a lot of interest in the post. We're working on a couple of other things. We want to be recognised as a centre of excellence, and there are ways of doing that. As I said, there are lots of good clinical studies ongoing in Dublin, patient collections in lots of the major rheumatic diseases. I'll show you this in the next slide. But really to continue to develop that, because clinical research and the research that I was talking to you about, ways of predicting who'll get severe disease, whether it's rheumatoid, psoriatic, arthritis, etc. We need to have well-characterised patient cohorts, and we need to be able to get samples that we can do experiments on, whether it's DNA or it's to do cells, and look to see how things are different between different patient groups. And that's key to doing what we call translational research. And so there are a couple of major strands. Autoimmune rheumatology is rheumatoid, ankylosing spondylitis, psoriatic arthritis, the primary inflammatory types of arthritis, very common. Lots of good work has been done over the years in Dublin. Rheumatoid arthritis, psoriatic arthritis, crystal arthritis. We're strong in crystal arthritis. Geraldine McCarthy works in the Mater and UCD, is a world-renowned figure in that area. And osteoarthritis. And we've got lots of strengths in different areas of technical areas, so in genetics, immunology, as I mentioned, imaging, very good in Dublin as well. David Kane, very strong in imaging. So this will all contribute, I think, and bringing things together and getting people working together is a key thing and a key objective. But you know, it's worked. Everyone seems very keen to do it, and it has been over the years. So hopefully that this will continue, and we'll build on a great base. So the sort of in the medium term, I want to extend it outside Dublin clearly, and bring in Cork, Galway, and Belfast actually as well into these studies, and getting recruiting patients to studies on many of these common conditions, rheumatic conditions. Develop research programs in those key areas that I mentioned, showed you in the last slide. Crystal arthritis, for example, osteoarthritis. Have to attract funding from various sources, not just Arthritis Ireland, but Wellcome Trust, European Union, etc., etc. And we need to get training for young researchers, different areas, medical, nursing, allied health professionals. The key stakeholders, and the key stakeholder is actually the patients. It's the patients will help drive this, and we'll have to really, and we'll have to talk with Arthritis Ireland about involving patients in research decisions. So, I mean, it's just key to do anything now. So I was in a committee in London on drug regulation today, and we have patients on the committees that make decisions on drugs, for example, and the regulators, whether it's the MHRA in England or it's the European Medicines Agency, always a patient's representative saying what they think of new drugs. Are these required? Does it look good? Did the drug company do the important measures of response, for example? And so patients actually now are driving lots of things, and we have to involve patients in our research and in getting ways, the patients telling us what areas they feel would be important to do research in. Okay, so that's important as well. So in Sheffield, I'd set up what we call in England was patient-public partnership committees, PPIs. Certainly in Sheffield, we set up. We just set one up in rheumatology, and patients would be involved in, for example, looking at research proposals, looking at ethics proposals, advising us on what I just said, on what they feel might be important areas that are under-researched. So this is another key area that we'll have to work on. So that's it. I hope I stuck to time. And are there any questions? Here comes John.
Gerry, that was absolutely fascinating. Did everybody enjoy that? Yeah. If I'd known you were going to do all that, I would have bought 2 of you. But that's absolutely fantastic, and I know lots of people in the audience are looking forward to us reporting back on that and keeping you up to date with what's happening, and I'm certainly looking forward to, and all the team in Arthritis Ireland are looking forward to working with you. Is there anybody, I'm sure there's lots of questions and statements or whatever, so let's start. There are 2 mics roaming up and down either side, so just put your hand up if you've got anything to ask Gerry. Now's your chance.
I was wondering, are you proposing setting up a register in Dublin for biologics?
Hope to set up a register for biologics in Dublin. Yes, I mean, I think it would be great if we did that. I'm not quite sure how that could be done. Doug, do you want to say, is that feasible?
The ISR did look at it a couple of years ago, and there were a couple of issues, particularly around the logistics of collecting that sort of data over different centres. Now, because we didn't have the wherewithal at that point in time, we didn't proceed with a national database, but we did with what we did was actually we set up a St. Vincent's database because we did have the capabilities there on site to do it. So we do have 1,400 patients in St. Vincent's who were entered onto a registry when they started on biologic therapies, and that data is there. And we do have, we can relate that to some of the other things that you mentioned in terms of the biological samples, the biobanks that we have as well. So I think it is feasible. I think there are certain logistics, and we obviously, as you said, we need everybody on board in a sense from all the different centres. But I think we've made a start, and hopefully we can progress it from there.
Yeah.
You mentioned imaging there, and you're talking a lot about X-rays in your research.
Yeah.
Ultrasound, and did you mean ultrasound?
Yes, ultrasound and MRI. It depends which they're different.
You were talking about Dublin being good.
Yeah.
And I attend rheumatologists in Dublin, and I asked about ultrasound, and I was told, well, we don't do that here. So I'm just wondering, when you say Dublin's good on ultrasound.
Yeah. It's good, right? It's good for academically for working out the roles of ultrasound. This is a general thing. I have to say, in Sheffield, we had very poor access to ultrasound, and a lot of places don't. But we really have to get that right. And you are trying to correct that and have standards of treatment. And ultrasound should really be there, but it isn't, and it's not just in Dublin or Ireland. As I say, in England, it's in lots of centres. It's difficult to get ultrasound, but it really has to be there because it's a great way of diagnosing subclinical inflammation.
The title of the talk was Working on a Cure. I was just wondering where are you at now, or where is anybody at in relation to that aspect?
Yeah, it's sort of interesting. I think the big thing that we realise now is when we essentially what we did in medicine over the years is to lump everyone together, okay, and just as a disease, okay, as a condition, as rheumatoid arthritis or ischemic heart disease or diabetes or blood pressure. And what we do these days is we actually try and split it apart because we realise there are completely different mechanisms that underlie the cause of that. And you can think about blood pressure. You can get blood pressure from a whole lot of different causes, from kidney disease, from family history, from poor diet, no exercise. Lots of things can feed into that. It gives you the final thing, which is blood pressure, but the way you get to that may be completely different. And so what we're doing now is to split things apart. And in that way, I think we might get to more potentially two cures, but I think it will be different cures for different patients. It'll be quite specific, okay? And so it's not going to be one thing for ankylosing spondylitis or one thing for rheumatoid. It'll turn out to be different things in different subgroups of patients.
While the use of injections has been very beneficial for a number of patients, what about where are we in terms of looking at oral drugs, where we can do away with the injections?
Yeah, that's a great question. So the biologicals that we use at the moment are really big, and that's why they have to be given by injections. So most of the tablets patients take are much smaller. So this, what we call the molecular size of an anti-TNF agent, is about 50,000 units. Any tablet that you take orally is less than 350 because it can get through the gut. Okay, you can't give it'll be broken down, a biological in the gut, and it can't do that. So that's one of the reasons why it is sad that the biologicals are expensive or are so costly, is because they're expensive to produce. So there have been a number of drugs that have gone through quite a lot and come into patients and study small molecules that can be taken orally. The trouble is they seem to have quite a narrow, what we call a therapeutic range. So what's effective is quite close to being what's very toxic. Yeah, you know, and that's the downside. The anti-TNFs are really very specific. They only hit one thing in the body. You know, you've got 30 or 40,000 different proteins being in the human body, and anti-TNF, it only hits one thing, and that's TNF. It picks up, it doesn't interact with the other, you know, 39,999 proteins. It only hits TNF. The smaller molecules are the ones you take orally seem to be much less specific, and they do things to things that you don't really want them to do. And so some of them, for example, they knock down the immune system to such an extent that the rate of infections is very high. And if you lower the dose, then the X-ray damage, you don't protect patients against developing X-ray damage. So it's a big difficulty. Now there are new areas, I think, that are, and drugs are coming out in new areas that target sort of the DNA and things in the DNA, which look to be potentially that they might work in inflammation. But again, you have to get these studies done in human populations just to see how effective and how specific they are. Because the good thing about the biologicals is that they're very specific. They only hit one thing, and that's why the side effect profile is really very good, considering how effective they are. Okay, corticosteroids are great drugs because they knock down inflammation, but they knock down lots of other things as well, and that's why you get lots of side effects. Whereas TNF hits one thing and doesn't have, and it's very specific, and so the side effect profile is very good.
Thank you. I'm just wondering, I suffer from various forms of arthritis, and my perception is when I go to my clinics and when I came here to have my exercise regime, the majority of us were women. And I just wonder, is that something that you come across? I can trace back arthritis in my family to my great-great-grandmother. And is that something that you know, you have come across, or is it me just being, you know, not perceiving maybe that there were many men there or whatever?
Well,
Thank you.
well, as a geneticist, I'd like to get your family DNA, I think, because you do learn a lot from individual families, and that's another thing I'd like to do, because the technology is going ahead. So, you know, most of these conditions, well, any of the common medical conditions, whether it's cardiovascular, it's lung, or it's arthritis, involve, we've maybe got about 100 genes now that increase the risk for rheumatoid, but they only increase by a little bit. But in some families you do see what we, you know, inheritance that's much more classical, And I suspect in those families there is probably a single quite dominant genetic cause. And we're getting close to be able to sequence the whole human genome in individuals for a couple of €100. And I think these sort of studies will be done in the next 5 to 10 years. And I think we'll find a lot of interesting causes. Coming back to your question, that for a subgroup of patients we'll find a genetic cause, particularly if there's a strong family history, multi-generation, and we will find particular rare causes. And so that is another thing. It's interesting, working with patients. You see these things present and people have phenomenal family histories. Whether it's, I've seen it with paraphosphate arthritis, and you can trace it down in a family, and I'm sure it's just due to a single gene, and that gene actually, it will tell you important things. You may not know that gene's involved in anything, for example, and this will tell you that it's important in the immune system or how in other aspects of biology. So I think that's a very good question, and I think it's another area that will be very productive in the next 5 to 10 years.
I'm just interested to know, has there been research done into the lifespan? All these new biologics and all the new drugs that come on stream. Is there a lifespan with them? Because I have found talking to people that they only get a certain amount of time out of them.
Yeah.
So what actually happens? Do we keep moving on to all the new drugs that are coming out? I've been on 3 or 4 or 5, maybe 5 so far, and I find the lifespan is quite short.
Yeah. I mean, it's sort of probably variable between patients. Some patients seem to be able to stay on in the long term. I think that you would agree, Doug. And other patients, move. And why that is, we don't know. Whether the inflammation becomes independent, for example, of TNF. That used to be said, that this inflammatory cytokines became less important as the rheumatoid was more established and you had to use other things, so it became independent of those. So it could be that in some, or perhaps developing antibodies against drugs, for example, is another issue.
You actually often initially started people on rituximab.
Yeah.
Now, for 6 months, people are often left on for 6 months at a time. But a lot of damage, if it doesn't actually work, a lot of damage can happen in that 6 months, which did, myself too. And I found it was a long time to be waiting for something to actually work.
Yeah.
And at the end, it was like taking Smarties. I was back to square one, couldn't get up and down the stairs, couldn't drive, nothing. And that was only recently, that was only in December. So I just find that's a long time to leave something.
Yeah. Yeah. So I'm just coming from England with NICE, and that's the yeah. I know. I mean, look. So lots of the funding agencies are really interested in working this out and trying to get the right drug as soon as possible. It doesn't actually save money, in a sense, to the drug budget, because patients are on the drugs. You're started on one, but you continually have to change. But what it does clearly in the longer run, it has benefits to you in terms of lifestyle, to the government in terms of health of the population, working, etc., etc. So it's an area that is important. It's quite variable between patients. It's a bit of what I was sort of saying. Everything is variable, and we're, as I was saying, probably the thing to remember from tonight is we don't lump everyone together anymore. We sort of split people apart, and science will allow us to look in the individual, okay, and try and come up with ways, whether it's a strong genetic cause or it's an environmental cause or whatever, but it's in the individual.
Just connected with that, to do with the biologicals, the anti-TNFs and the interleukin-6s, you're saying that you're testing people for the CCP gene, which is whether it's positive or negative, and hopefully being able to give the person the right drug. So at the moment, now I know you're only arriving here, so I don't know whether you'll give answers.
No pressure, Gerry. No pressure.
No pressure.
But it's actually, I've done research on it, and it's
Yeah.
also in England and in the States. You have to have tried the TNF drugs before you can go on to the interleukin-6 drugs.
Yeah.
And I'm wondering why.
Right. Well, these things are evolving, and so the recent paper, there's a paper in Lancet about 4 months ago showing that anti-IL-6 was actually, if you look at, they compared a couple of hundred patients starting Humira with anti-IL-6, and IL-6 is actually superior. So it may well change. Okay, so it's all about the psychology of how we use drugs. I think, TNF is there first. It doesn't mean it's the best. It means that everyone's got most experience using it. It's the same for lots of things, why we use drugs. It's just because, I sit in clinic, I maybe know the dose for 4 or 5 non-steroidal anti-inflammatories, well, probably 3, but there are 30 or 40, so I use the ones I'm familiar with. And that's just,
It's policy in,
Yeah.
I think, all the hospitals here, that you go through your Humira, your Enbrel, your Cimzia, your Simponi, before you're allowed
Is that right?
to go on to.
That's correct.
Well, okay, so the hard evidence for this, and Doug, you'll tell me if I'm wrong, so the rate of non-response is about 35% for an anti-TNF. But if you go on to a second anti-TNF, the rate of non-response is about 50%. So in Sheffield, I drawn up our protocols for using the anti-TNFs, and we were just about to change it so that we'd only use one anti-TNF. So, okay, so what we had previously done were 2 anti-TNFs, either Enbrel or an antibody, as 1 or 2, because they're quite different in structure and what they do between the antibodies and Enbrel. Okay, and it was up to the patient to decide whether it was this pen or whatever, but the patient made the decision. If that failed, then they would go on to the other type of anti-TNF, okay? And then the third line was based, if the patient was CCP positive, it was Ritux, CCP negative, it was IL-6. That's how we did it. We'd stopped doing that, and actually the guidelines which we're probably now using in Sheffield is just 1 anti-TNF.
Just to clarify that, actually,
Okay.
the reason tocilizumab actually was decided in most hospitals to be 3rd line was because it's given as an IV therapy in the hospital, and the hospital then has to pay for the cost of the drug.
Okay.
And that's why it was positioned in Ireland in that fashion. But in fact, if I might just make it a small announcement, we're just about to start a study of tocilizumab as a subcutaneous injection, which patients can get, like Humira or one of the TNFs.
Very good.
So that study is going to be starting in about 4 weeks' time. So
Excellent. Watch this space. We'll have you back here in 4 weeks.
any volunteers?
Give him 6. 6, okay then. We'll take one more question, just in the interest of time. Very few people are aware that Gerry's plane just landed at 6:45, I think it was, so that's marketing for you. Any more questions? Just one more here? Okay, we've 2 more then.
It's just in relation to other inflammatory diseases, not rheumatology. They're working on similar type drugs, if you like, the Remicade and the Humira and all the rest of it. Are they doing the same type of research here in terms of TNFs and all that stuff?
So you mean other areas like inflammatory bowel condition?
Well, also colitis and that.
Yeah. So I presume they are, but they use a much more limited spectrum of biological agents because, for example, Enbrel doesn't work. So it's only the antibodies, TNF, that work. And I don't think they don't use Ritux and they don't use anti-IL-6. So it's the revolution has been in rheumatology. We've been very lucky that it's really been rheumatology, and it hasn't extended to, for example, inflammatory neurological conditions. Because as you know, anti-TNF can exacerbate things like MS, which is an inflammatory condition, which is many of the genes that cause rheumatoid are important in MS as well. But anti-TNF doesn't, it makes it worse. So for inflammatory joint disease, in many ways we're lucky. that for various reasons we've sort of hit upon a large number of targets. And part of that is because I think animal models, but also because rheumatologists and patients have, we've been able to get samples of tissue from patients, from joints, and do experiments. We've got great animal models of inflammatory arthritis, and they've really been very useful in working out what's important. And that goes back 20 years. Someone 20 years ago, I think it was this month, where they took a mouse and they put the human TNF gene into it and just switched it on continually, and the mouse didn't develop psoriasis or MS or a ton of other things. The mouse developed rheumatoid arthritis.
I would like to thank you all for coming and taking the time out. I think we could go on a lot longer, because certainly for me this has been one of the most interesting talks we've had within Arthritis Ireland. I'm, as a funder and responsible ultimately for spending donors' money, I'm greatly encouraged by the fantastic collaboration between UCD and TCD. And really, both institutions are talking about investing in rheumatology for the country. And it's very, there's really no territory talk going on at all. It's actually really, really encouraging, and we're very encouraged from a patient support organisation. So a big thank you to Gerry Wilson for coming here tonight. So we hope to bring you many more of these talks. Not in 6 weeks' time. We'll give you a little bit more time, Gerry.
Doug 6 weeks.
Doug 6 weeks, yeah, okay. Okay, well, thanks again, and safe home.
Thank you.